is the sarcolemma, the actual cell membrane. So what's this connective tissue layer here surrounding this one cell? You should know this one by now. The innermost. What's the innermost layer of connective tissue surrounding individual muscle cells? Is it fascia? Is it epimysium? Is it perimysium? Or is it endomysium? Endomysium connective tissue surrounding individual cells. Okay? By the way, capillaries would run through the endomysium, which they're not bothering to show here. We do see the distal end of one axon collateral of one somatic efferent neuron. So you can see the node of Ranvier, Schwann cell with producing a myelin sheath. This cross-sectional view shows the myelin sheath around the axon here. So again, one axon collateral leading to this one muscle cell. So this rippled region of the sarcolemma, this is the cell membrane of sarcolemma. This rippled region of the sarcolemma is the motor end plate. What's significant about the motor end plate? That's where all the nicotinic cholinergic receptors are located to bind acetylcholine molecules. So there's a synaptic end bulb, lots of vesicles inside there packed with acetylcholine. As the potential gets to here, you would see the secretion of acetylcholine onto the motor end plate, and the molecules of acetylcholine would bind to the nicotinic cholinergic receptors. So why the ripples? Why the folds? Folding increases surface area. So you've got this patch of membrane, highly folded, but you can pack in many, many more nicotinic cholinergic receptors. The more folds, the more receptors you can pack into those folds. Ain't nature grand? All right, what else do we have here? This cross-sectional view of this skeletal muscle cell. Every brown dot you see, remember this is one myofiber. This is a model of one cell, one myofiber. Every myofiber contains many myofibrils. Every brown dot you see is a myofibril. Okay? In cross section. These gaps you see in here don't really exist. That's another mistake. So they are really packed in there. Myofibers are packed cheek by jowl within the sarcoplasm. So a longitudinal view reveals the myofibrils running lengthwise. So we see about 20 myofibrils running lengthwise here. You notice that they're dark brown, light brown, or dark brown, beige, etc. That's because every myofibril is divided up into sarcomeres. Remember, sarcomere is the smallest contractile unit of a muscle cell, extending from one Z disc to the next Z disc. So, where would that be? See this skinny black line running all the way through? That's a Z disc. The next skinny black line, the next skinny black line, the next skinny black line, the next skinny black line. Every skinny black line running throughout the cell is its own separate Z disc. Okay, so so here we see from Z disc to Z disc. So this portion of this myofibril from Z disc to Z disc, this is one sarcomere there. From this Z disc to this one, here's the second sarcomere. From Z disc to Z disc, here's a third sarcomere. And under it is another sarcomere. Under it is another sarcomere. Under it is another sarcomere. So it's a section, one myofibril from Z disc to Z disc. That's the smallest contracting unit of the entire cell, right there, like a microscopic accordion, in fact, when it contracts. Okay, so what's in the center of a Z disc? See this little brown rectangle there, this little brown patch? That's called an A-band.